Glycolysis. Gluco means sugar and lysis means split or breakage. This pathway is also known as Embedment-Markov pathway, a major pathway of glucose breakage. In this pathway, the oxidation of glucose takes place. It is a primary pathway used by all organisms. It can be in presence or absence of oxygen. The end product of glucose in this pathway is two pyruvic acid molecules. One molecule of glucose gives two pyruvic acids. Glucose enters glycolysis by phosphorylation to glucose 6-phosphate catalyzed by hexokinase or glucokinase by using ATP as the phosphate donor. Under physiological conditions, the phosphorylation of glucose to glucose 6-phosphate can be regarded as irreversible. ATP converted into ADP. Glucose 6-phosphate is an important compound at the junction of several metabolic pathways: glycolysis, glycogenesis, and glycogenolysis. In this reaction, glucose 6-phosphate is changed into fructose 6-phosphate with the help of an enzyme phosphohexokinase isomerase. In this step, fructose 6-phosphate is converted into d-fructose 1,6-bisphosphate and other phosphate group attached at carbon position number 6. The source of phosphate group is ATP, which is converted into ADP. An enzyme takes part in this reaction called as phosphofructokinase. In this step, the cleavage of d-fructose 1,6-bisphosphate takes place. There are two products of this step. One of them is dihydroxyacetone phosphate with the help of an enzyme aldolase, and second one is glyceraldehyde 3-phosphate. In this step, dihydroxyacetone phosphate is changed into glyceraldehyde 3-phosphate with the help of an enzyme phosphotriose isomerase. Continues with the help of oxidation of glyceraldehyde 3-phosphate to 1,3-bisphosphate phosphoglycerate. The enzyme catalyzing this oxidation, glyceraldehyde 3-phosphate dehydrogenase, is NAD dependent. This reaction, catalyzed by phosphoglycerate kinase, phosphate is transferred from 1,3-bisphosphoglycerate into ADP, forming ATP, substrate level phosphorylation, and 3-phosphoglycerate. In this reaction, 3-phosphoglycerate is changed into 2-phosphoglycerate with the help of an enzyme phosphoglycerate mutase, which catalyzes this reaction. The subsequent step is catalyzed by enolase and involves dehydration, forming phosphophenol pyruvate. Enolase is inhibited by fluoride and also dependent on the presence of either magnesium positive 2 or manganese positive 2. The phosphate of phosphoenol pyruvate is transferred to ADP by pyruvate kinase to form 2 molecules of ATP per molecule of glucose oxidized. The product of this reaction is pyruvate enol. Method of ATP formation: respiratory chain oxidation of 2-NADH6, substrate level phosphorylation 2, substrate level phosphorylation 2, total 10, consumption -2, net ATP formation is 8 molecules of adenosine triphosphate, means ATP.